Now let us understand the structure of the nucleic acids. See, the nucleic acids, as I've told you, are polynucleotides. Their simple monomer unit is a nucleotide, which is made up of three types of molecular species called nitrogenous bases, pentose sugar, and phosphoric acid molecule. Let us first see a sugar molecule contains a nitrogen base attached to its first carbon atom. And the combination of a nitrogen base and pentose sugar is called a nucleoside, SIDE. Now, when a phosphoric acid molecule combines with this, it attaches itself to the fifth carbon atom of the pentose sugar like this. The OH of this alcohol of the fifth carbon and the OH group of the phosphoric acid, they loses water, and this is how they get attached. Now, this combination of phosphoric acid, nitrogenous base, and this pentose sugar, the phosphoric acid, nitrogenous base, pentose sugar, these three is called nucleotide. Now, the nitrogenous base is always present at carbon number 1 of the pentose sugar, and the phosphoric acid is attached to the carbon number 5. Now, how these nucleotides gets bonded to each other? Let us see this. They get bounded to each other like this. This is the nitrogenous base there on the first carbon atom, and this is the third carbon atom's OH group. Now what happens is, what we find is, this is the second nucleotide we have. This is one nucleotide. This is again is the one which is having what? Phosphoric like this. Now what happens is, this phosphoric acid molecule gets bonded through this common oxygen with this carbon number 3 of the other nucleotide. So what happens is, like this, one nucleotide gets bonded to the other nucleotide through phosphoric acid molecular species, and what this phosphoric acid molecule is linking the carbon number 3 of the one nucleotide's pentose sugar with the carbon number 5 of the other nucleotide's pentose sugar. And this type of linkage which this phosphoric acid is forming between the two nucleotides is called phosphodiester bond. This bond is called phosphodiester bond, and this is the one which is responsible for connecting the nucleotides. Now it becomes dinucleotide. Similarly, one more unit will, if it combines, becomes trinucleotide, and slowly the chain is formed which is called polynucleotide, which can be described in a very simple manner like this. So this is the sugar. We have phosphoric acid, sugar, 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 phosphoric acid, and sugar. So this is how the chain goes. It starts from a sugar, ends at a sugar, linked to each other through phosphorus, and each sugar is having a nitrogenous base, maybe the adenine, a guanine, cytosine, thymine, adenine, thymine, guanine, cytosine, whatever it is. Now this sequence of the nitrogenous bases on the chain of these nucleotides, which is called polynucleotide, is called what? Genetic information, or it is called the genetic information which is there, which is stored in these polynucleotide chains in the form of the sequence of these nitrogenous bases. Now the combination of these three is called what we call as codon, and this is the one which is responsible for the genetic information. Now these polynucleotide chains are the one which are the main structural material of the nucleic acids. And let us see how they exist in the DNA and RNA. Let us first see the DNA. Now the structure of DNA, that's most important to understand. Now let us understand the structure of the DNA and RNA. See, the nucleic acids, what is the exact structure is, I have explained it to you. But actually these polynucleotides exist in the form of the secondary structures in the form which is called DNA or RNA. So DNA's and RNA's structures is explained by what which is by the structures which are called secondary structures of these nucleic acids. Now, DNA's secondary structure is a double helical structure. It was given by Watson and Crick, and it's called Watson-Crick model. Now, DNA consists of a double helical. It's a two polynucleotide chains, so it's called double helical, and these two chains of polynucleotides are opposite or anti-parallel. Like if this chain is starting from the third carbon, it will be ending at 5, and similarly if it is starting from 5th, it will be ending at 3. So they are anti-parallel, and the two chains are bonded to each other through hydrogen bonds through their nitrogenous bases. Adenine is always forming pair with thymine through 2 hydrogen bonds, and guanine will be always forming 3 hydrogen bonds with cytosine. So that is how these nitrogenous bases are always complementary, that adenine is always bonded to thymine through 2 hydrogen bonds, and guanine is always bonded to cytosine through 3 hydrogen bonds. And that is why we say that the two polynucleotide chains in DNA are complementary to each other. So if we know the sequence of one of the polynucleotide chain, we can exactly know the sequence of the nitrogen bases in the second polynucleotide chain in the DNA. So this is how this helical structure of the two nucleotides makes the DNA model or secondary structure of the DNA. Now let us move to RNA. RNA is not double helical. The secondary structure of RNA is consisting of a single polynucleotide chain in which we have what sugar phosphorus sequence chain, and the sugar molecules are having nitrogenous bases, which is actually the sequence in which the information is transferred. Now RNAs are of 3 types: mRNA, rRNA, and tRNA. mRNA means messenger RNA, rRNA means ribosomal RNA, and tRNA means transfer RNA. They perform different functions, that is why they are of 3 types. Now, after understanding the structure of and how the nucleic acids the construction of the nucleic acids basically from the molecular level, now let us move to the biological functions of the nucleic acids. See, nucleic acids perform 2 major biological functions. The first is the transferring of the genetic information from one generation to the other generation. Whenever the cell is about to divide, what happens is the DNA duplicates. One more DNA is formed, so that both the daughter cells are getting one set of the DNAs. Now this division takes place because the genetic information is transferred to the next generation. Now this is called replication. So the most important function of the nucleic acids is to transfer the genetic information from one generation to the other generation. That's the most important function. The second function is the protein synthesis. The nucleic acids controls the traits or the characters of a species or of the organisms through the protein synthesis. How or which type of a protein is to be synthesized in which organism and where, this all information is stored in these DNAs in the form of the sequence of nitrogenous bases, which is called codon, or the codes, which is the sequence of these nitrogenous bases. Now these nitrogenous bases are actually being transferred. The information is transferred to messenger RNA, which carries this information from the nucleus of the cell and moves out, and with the help of ribosomal and transfer RNA, it synthesizes a particular protein which performs a particular type of function in the body. This is how the other most important function of the nucleic acids is performed. That is the protein synthesis. Because if those proteins are not synthesized, those specific functions of growth and development will not be done. This is how the 2 major important functions of biological functions are performed by the nucleic acids.